All right, I'm really excited to be giving this webinar, and as Mohammed pointed out, I hope that afterwards, whoever's listening out there who's not familiar with microbes and the role that they play in our society and in our environment will have a better idea of what are microorganisms and what are they doing. So part of the reason why microorganisms have historically been overlooked for many centuries is actually due to their small size. In the microbial world, you have a lot of organisms that belong to several different groups. We often hear about bacteria, of course, because they're involved in a lot of pathogenesis in humans, but actually in the environment and in human beings, you can also find another group of organisms known as the archaea. We also have viruses, protists, and fungi. And if you look at the scale, you can actually see microorganisms are very small. If we compare them to, for example, the human blood cell or salt, we can see that they're very tiny organisms. So where do they stand as far as the food chain is involved? If we look at the food web in our environment, we see that they're actually at the bottom of the food web. And they're very important for the foundation of our aquatic and terrestrial ecosystems because of this. So for example, we have predators at the top and we have secondary and primary consumers, and all of these organisms at the top of the food web rely on microorganisms in order to survive. And that's true in both aquatic and terrestrial systems. And when we look at where they stand in general, evolutionarily, when we look at the history of our planet, we see that the first evidence of microorganisms appears around 4 billion years in comparison to humans. They were around way before our atmosphere formed as we know it. When we compare the diversity and ancestry of microorganisms, we see that they are found in more than one group of life or domain of life. And the current theory out there is that the archaeal and bacterial branches of life actually gave rise to eukaryotes, to which we humans belong to. And furthermore, if we look early on in time, we can actually see that we shared a common universal ancestor. And this is called the LUCA, or last universal common ancestor. So microorganisms are very old, they are very diverse, and they are many. So where do microorganisms come from? Where are they found? Microorganisms inhabit a variety of habitats, including the human stomach. They can also exist in soil, the ocean floor, and more extreme habitats, such as hot springs, caverns, deep sea hydrothermal vents, and frozen soils. Now, in our society, microorganisms play an important role in the food industry. For example, we get our cheese and our beer with the help of microorganisms, and the industry readily exploits them to give us some of our very delicious foods. We also use them to clean up our waste. And examples of this can be heard of in the news. So for example, in the Gulf oil spill. Or in another example is the Oak Ridge National Lab in the United States, where microorganisms are used, for example, to bioremediate uranium contamination. So microorganisms can also affect human health. So for example, we frequently hear about people getting food poisoning from contaminated foods. So in a sense, we're living in a microbial world. So if we look here, we know that a small percentage of microorganisms are disease-causing, but a lot of them actually are involved in controlling and driving environmental processes. So they can do a lot of things, like they can breathe on minerals, on gases like methane and nitrogen. They can help us break down organic matter, help us cycle nutrients and chemicals, and they do this on ocean and land. And so microorganisms really are important in helping to drive a lot of Earth's cycles, including the nitrogen, carbon, sulfur, phosphorus, and iron cycles. So in the next couple of slides, I've highlighted some key discoveries that have paved the way for microbiology today. So for example, in 1665, we have Robert Hooke, who publishes Micrographia, and in here, in this book, he details the observations he's making with fossils, shells, and living things with his microscope. Then in 1677, Anthony Leeuwenhoek discovers microorganisms using his microscope and reports these observations to the Royal Society. By 1862, you have Louis Pasteur, who develops the germ theory of disease. And by 1881, we have Robert Koch, who discovers how to actually grow bacteria on solid media. And ever since, actually, we've been using this technique. By 1887, we had R.J. Petrie, and he was the inventor of the Petri dish. And this, in combination with Koch's discovery, has helped us in order to grow bacteria and other types of organisms. By 1892, Dmitry Ivanovich discovers viruses. And by 1928, Alexander Fleming discovers penicillin. So a field more and more recognized today is that of geomicrobiology, which has brought together the fields of geology and microbiology in order to understand how microorganisms can influence the geology and chemistry of an environment and vice versa. Early on in the 1920s, Edson Baskin, who's a geologist, and Frank Greer, who were the microbiologists in the US, discovered a group of organisms that could help cycle sulfate in a deep oil field. And this is actually considered to be the origin of geomicrobiology. However, this discovery was dismissed for over 50 years. And it wasn't until 1987 that the US Department of Energy was drilling a borehole. And they discovered that there were microorganisms living very deep below the surface. And this actually confirmed the findings of the 1920s. So shown on the lower left-hand panel is a picture of Rio Tinto in Spain. And this is a site of mining activity. Now, the waters of Rio Tinto are very acidic. They have a very low pH of 2, and they're very high in metals. Now, this low pH is mainly due to the activity of microorganisms. In the middle is a picture of Lost City, which is a hydrothermal vent system located near the Mid-Atlantic Bridge. This vent system was discovered in the 2000s, and it was found to harbor microorganisms that survive on hydrogen and methane, as well as basic conditions. And pictured on the right is one of the many hot springs at Yellowstone National Park. So a lot of microbes also inhabit our urban environments, and that's something that I've actually been thinking about more and more. They exist in our transportation areas, our public areas, soils, gardens, park, groundwater, our homes, our pets, our bodies. So the picture on the left is one of the Chicago City Hall building in the US. And recent studies have focused on understanding how microbes inhabiting, for example, the green roof environment, which is the one pictured there, in urban areas might differ from those that exist outside in the typical natural environment, and what are factors that play a role in their distribution and diversity. Certain microbes in our urban atmosphere, they're transient. They can be transported between urban environments, or they can actually be transported across the oceans from other continents during storms. And you can see that in the middle picture. This is a NASA image that was taken of dust that is traversing the ocean. Another area of research in which bacteria are involved is antibiotic resistance. Now, in nature, antibiotic resistance does occur, even in caves that have been isolated from humans for millions of years. However, the overuse of prescription antibiotics by humans has actually given rise to a lot of microbes that are resistant in the environment. There was a recent article in Scientific America that was actually covering this research that was published in Scientific Reports, and it highlights that general public areas in London can actually be reservoirs for multi-drug resistant bacteria. So this is just one of many examples of how humanity has played a role in influencing the evolution of microbes. As I mentioned before, microbes are more and more being studied in the urban environment in order to understand how they could differ, perhaps from other more remote areas. A study published in 2014 was conducted by Kelly Ramirez and her collaborators, and this focused on characterizing the microbial diversity present in New York Central Park. Now, they analyzed over 500 samples of soil and found these microbes to be as diverse as those in other biomes, including the Arctic, desert, and tropical soils. So the majority are not well studied, as is the case with many microbes from the environment, but this discovery was very exciting. So a lot of microbes that inhabit soils and green roofs in the urban environment, as I mentioned before, help to cycle a lot of our elements. And one example of this is a recent publication that focused on nitrogen cycling organisms in green roof ecosystems. Now, this book here, Never Home Alone, actually had a huge impact on me this summer while in vacation. I really recommend this book, which you can find on Amazon by Rob Dunn. And it really made me think about this concept that microbes can be studied actually in our urban environment and in our homes, and how important scientific discoveries have actually come from looking at microorganisms that inhabit our homes. So one of the studies that I will highlight from this book, along with two others, has to do with microorganisms that are found in the showerhead of many homes in the US. And this was a study that was made by Cecil, who basically looked at what kind of microorganisms are inhabiting the showerheads of people in the US. And what they found was that mycobacteria make up about 26% of the biofilms that are being formed. And I'm going to go now into a little detail regarding that. I also want to talk a little bit about some of the microbes in our kitchen and how great discoveries have come from looking at the microbes that inhabit insects in homes. So studies by Noah Fier and Rob Dunn focus on understanding the types of microorganisms inhabiting showerheads in the US. Through their studies, they found that the type of showerhead could influence the type of microbes that come to inhabit them and grow in a slime called biofilm, which is essentially a gunk that's made of many microbes that excrete their waste. And so this waste clumps together, and that's what we call a biofilm. The biofilm helps protect these microbes from the harsh conditions in the showerhead. Water from a source, such as a river or a well, is very diverse in the types of microbes that inhabit it. Water that is treated with biocides will reduce this diversity and also abundance. Now, biocides are used to make drinking water safe if it contains too many pathogens. Water from deep, old aquifers, on the other hand, are not treated, as this water tends to be naturally free of pathogens and is safe to drink. Even if water, though, is treated with biocides, you might not know this, but it still contains microbes, namely those that were not killed by the biocide. What Fier and Dunn and their collaborators found over the course of their studies is that different factors, including the showerhead model, the temperature, water flow rate, whether or not biocides were used to treat the water going into the home, the geographical location, and even the water source are all different factors that can play a role that influence what kind of microbes inhabit the showerheads. Specifically, they observed more mycobacteria in showerheads receiving water from municipal systems in the US versus well water. Now, the mycobacteria in showerheads included non-pathogenic as well as pathogenic species that can cause lung disease. So the authors hypothesized that the high chlorine levels used in municipal waters in the US may be a factor that selects for these types of mycobacteria. Now, further work was carried out by Caitlin Proctor, and she was a researcher at the ETH University in Zurich at the time. And what Proctor went on to further develop is to look at the biofilms that inhabit the shower hoses. And she looked at shower hoses in 11 countries. She found that potentially opportunistic pathogens were present in the houses she studied, including Legionella. And that the type of material that was used, so in this case, plastic tubings, appear to favor more biofilm growth than the metal do. Now, another study that was highlighted in this book by Dunn was how microbes that are used in baking can also play a role in how perhaps the bread tastes. So you might not know this, but for bread baking with yeast, bread can be baked using either store-bought yeast or yeast from a starter culture. And what's a starter culture? It's flour that is exposed to air. And the microbes that fall into the vessel are fed over several days to enrich for acid-producing bacteria and fungi. Now, the acidity produced by these microbes helps to keep pathogenic microbes from surviving, which is a good thing. So, for example, lactobacillus and Saccharomyces are common acid-producing microbes that are found in starter cultures. Now, one question that Rob Dunn and colleagues had regarding starter cultures was whether the microbes in the air of the bakeries and on the hands of the bakers would influence the starter culture microbial composition, and in turn, the flavor the bread baked with the starter culture would have. So it's known that starter cultures differ between bakeries, but at the time, no one knew why. So to try and answer this question, Dunn and colleagues performed an experiment in which 15 different bakers in 14 different countries were given the same ingredients to make the same starter culture. Now, their hypothesis was that microbes on the hands of the bakers and the home or bakery would influence the microbes in the starter. What they found was that the hands of the bakers were contributing to the starter culture. The other factor appeared to be the environment in which the starter culture was made. So they also found that different microbes were present in different regions, and they could show this by doing a global analysis of starter cultures. Now, the last study that I was really influenced with in this book had to do with insects and how insects commonly found in homes could harbor unique microbes that help to digest hard to break down compounds like plastics. So in a study conducted by Stephanie Matthews and collaborators, researchers set out to determine if camel crickets, which is an inhabitant in home and US, could harbor lignin-degrading microbes. Now, lignin is a molecule that makes up wood and paper products. Black liquor is a substance that is toxic and very basic. It is a waste product from the paper pulp industry that is commonly produced during the chemical breakdown of pulp wood. Before this study, there were only a handful of species of bacteria that were known to exist that could break down lignin. This study was able to show that from 3 greenhouse camel crickets and 3 hive beetles, 14 bacterial strains were identified that were capable of growth on lignocellulosic compounds, and the other one could grow with lignin alone. So this is an example of how microbes found in insects in a home could actually provide new information out there that the industry could then use. Now, in the ocean, microorganisms are known to influence various chemical cycles. Sometimes it's evident as physical structures, such as these rock-like structures pictured on the upper corner. These are called stromatolites. And these are layered beds formed in shallow water by sediment grains that are trapped by bacterial biofilms. And over time, they can become fossilized. Some of the oldest forms of life are found in fossilized stromatolites. Now below that is a picture of a living stromatolite, the different layers representing different microbial groups. Other times, the presence of microorganisms is not so evident. However, we have ocean expeditions that have allowed us to probe the microbial secrets of the deep far below the ocean floor. And one of those programs that does this is actually called the International Ocean Drilling Program, or IODP. Exploration and research of the world's oceans has shown that microorganisms carry out dynamic activities in the water column and in sediments. So pictured on the right, you have a picture of a sediment with different layers. And in the absence of oxygen, you can have different types of microorganisms that help cycle different nutrients that are present in these layers. Now, recently, there was a publication that's published by many scientists warning humanity that microorganisms are very much linked to climate change. The main point of this review, that's called the Microbiologist Warning, aims to raise awareness of the microbial world and make a call to action for microbiologists to become increasingly engaged in, and microbial research to become increasingly infused into the frameworks for addressing climate change. So one of the schematics that was shown is pictured here. You have the various environments in which microbes inhabit and help to intake and release carbon dioxide and methane, which are two greenhouse gases. Microbes in the ocean can intake carbon dioxide and in combination with the energy of light produce oxygen and sugars in a process called photosynthesis. When these types of microbes die, other microorganisms help to decompose them. Those microbes that help with the decomposition process release carbon dioxide into the environment when they respire in a process called remineralization. So this can happen both in the ocean and in terrestrial systems. Eventually, whatever isn't decomposed gets buried in the seafloor and over millions of years is converted to fossil fuels via geological processes. Similarly, in the terrestrial system, microbes can help decompose organic matter into carbon dioxide and methane through respiration and methanogenesis. Industrial processes, however, quickly release carbon dioxide stored in fossil fuels into our atmosphere. The authors of this paper go on to warn us that the many effects of human activities, including agriculture, industry, transport, population growth, and human consumption, combined with local environmental factors, including soil type, light, and life, greatly influence the complex network of microbial interactions that occur with other microbes, plants, and animals. Now, the last topic that I want to bring is one that has become increasingly of interest to me, and that has to do with electricity generating microbes. So microbes in the environment can also be used to generate energy for bioremediation purposes. One type of electricity generating device that is used to harvest energy from microbes that are present in the environment is called a microbial fuel cell, and that's pictured on the left. There are different types of microbial fuel cells. The one pictured here works by sticking an anode into the sediment while keeping the cathode on top of the sediment. The idea is that sediment-dwelling electricity-generating microbes metabolize the nutrients around them in the absence of oxygen, and then transfer electrons outside their cells to an anode in the sediment. Not all microorganisms can do these. These electrons are a byproduct of their metabolism. These electrons then flow up a wire that is connected to a cathode, which is sitting on top of the sediment, where they help power a device by producing current, in this case, a light bulb pictured in the cartoon. The electrons then travel back down the wire onto the cathode, and their electrons interact with oxygen and protons to form water. And then pictured here are also examples of electricigens, which are microbes that can carry out this process. And in this slide is just to give you an idea that these microbes can transfer electrons to the anode either by forming these gunky biofilms or with extensions that are called nanowires, or through a chemical mediator. And on the right-hand side is to show you that similarly, microbes can help to basically break down pollutants in the environment using a sort of microbial fuel cell as well. So briefly, before concluding this webinar, I wanted to reiterate a little bit about myself. I'm actually originally from California, from Los Angeles. Most of my life was spent there during my bachelor's and master's studies. During my PhD, I moved up to UC Santa Cruz in order to complete a PhD in microbiology. And after that, I went on to receive an NSF award to study at the University of Bremen, where I learned more about microorganisms in marine sediments. And currently, I am lecturing at the University of Vienna. And just to highlight a little bit about my research interests, as Mohammed may have pointed out, in the past, I worked with some of these electricigens, Shionella, but in this case, to look at their metabolism and how they help to cycle iron. And then in Bremen, I went more in depth into looking at the microbes that are present in marine sediments and how those help to cycle iron. And during my Marie Curie fellowship at the University of Vienna, I studied copper uptake by archaea. And furthermore, I'm collaborating now on a project where I'm looking at how fungi can influence weathering of rocks. So hopefully with this, I have shown you how microbes can influence the environment and also society. And some closing remarks for this. For the first half of geological time, our ancestors were bacteria. Most creatures still are bacteria, and each one of our trillions of cells is a colony of bacteria. This was predictable. Microorganisms respond to chemical and physical changes in an environment. Everything that you do to your environment affects the microorganisms that live there, and vice versa. Bacteria represent the world's greatest success story. They are today and have always been the model organisms on Earth. But hey, I just talked about archaea, so don't forget those. They cannot be nuked to oblivion and will outlive us all. And with that, I'd like to acknowledge the Marie Curie Alumni Association Austrian chapter, as well as the main association itself. And I would be open to taking any questions. Thank you.
So, Carolina, thank you so much for such a nice presentation. It was an introduction to microbes and microorganisms, and it was quite like fascinating for me. Like these tiny organisms that are so small that we can see them without a microscope can have us they play such a great big role in our life. So I would like to open the session for questions. Thank you for sharing your webcam with us as well. So if anyone has any question that would like to ask, we are open to your questions. You can simply write the questions in the chat box, or you can basically raise your hand using the features in the GoToWebinar, and then I will unmute you that you can ask your question yourself. So until the time that the people probably think about any questions that they have, probably I can ask you one question regarding. So we understood that basically the microbes are important for our body and also for our environment. It is interesting for me to know, Carolina, if there are any important industrial applications that are already being already used in everyday interactions that we have with microbes in the world. You mentioned a few of these applications for the climate change and so on. But I'm interested if there are any larger scale applications.
Yeah, so in a lot of the industrial applications for them have been in isolating, for example, enzymes, which are proteins, not only with breaking down chemical compounds, but also in producing chemical compounds. So I talked a little bit of history and how penicillin came about. That's actually a byproduct of a fungi. And so microorganisms do contribute a lot to applied microbiology, I would say. I know that there are also, there's a company that's out there that is trying to use this microbial fuel cell concept in order to generate clean water in places where they don't have a wastewater treatment plant. This company is run by the CEO, Oliveira, and it's called Aquafil, I believe.
All right, all right. So, is there any other questions from you guys, Andrew, Bagish, if you have any questions? Otherwise, we will end the session and we will forward the presentation, the PDF presentations to all the registrants. So thank you so much, Carolina.
Yeah, well, thank you very much.
Such a nice presentation. I mean, it's so hard to, sometimes it's so hard to present like a very specified matter in a very general format that can be understandable like to the people that they have like no expertise in that field. So you did a great job. And
well, I was really happy to have that opportunity.
We would like to continue these sessions. We would like to have like every month an introduction to the field of research of different people, different members that we have in this series of the webinar. So I'm closing the session. Thank you so much, and have a nice and great weekend on Friday.